Hi everyone. So I figured I would give you some notes and answer some of your questions. So we began talking about genetics a little bit last week. Actually, before that, because we talked about those black and white twins. So we talked about how different characteristics, also known as traits, are passed on from parents to their offsprings, meaning their children. So some of you might look like your parents, some of you may or may not, and we'll talk later about why that is the case. But we're basically going to review a little bit of what we discussed, and then I'm going to give you some examples of practice problems. So in terms of some of the videos that I posted, right, Mendel, again, Gregor, not George, he was a monk who, I think he failed medical school or didn't want to go to medical school, and he decided to do some experiments with pea plants because they were very easy to breed and it was very easy to see different traits in them. So he looked at things like color and height and pea size and whether they were round or wrinkled, and it was very, very easy to pick out single traits. Now, some of the words that you will need to know for later, especially, you have your P generation, which is your parents. You have your F1 generation, which are their offsprings. And then you have the F2 generation, which are the following offspring. So if you're thinking of these are your parents, this is you, and then these are your kids or your parents' grandkids, right? And as you can see, he took tall plants and short plants. He crossed them and he didn't see intermediate plants. He actually saw only tall plants, which he thought was strange. So he wanted to know if he would see the same thing in the following generation. So what he basically did was he took these plants, technically brothers and sisters, um, and he crossed them together. So he took the pollen, which is the sperm from one plant, and he put it on the other plant, and so forth, and fertilized the egg and basically made the new generation, the F2s. And what he saw was that you see the short plants reappear again. So that's basically how some of the hidden traits that you might have reappear. So maybe you have two parents and they both have brown eyes, and you have blue eyes, or maybe you have a family member with blue eyes, and that's basically how you would see a trait appear in the next generation that's not seen in the parents or sometimes even the grandparents. So in terms of some of Mendel's conclusions, and again, you should be familiar with these words at this point, you heard them at some point, right, is that there are dominant traits, which he called strong traits. In class, we talked about how those are the traits that will be present if they're found, right? So if the plant has at least one capital T, it will be a tall plant, right? In order to show the recessive traits, you have to have two of the lowercase letters, right? So these are your short or dwarf plants, and you hopefully saw those in the examples that you were working on. So in terms of more vocab, again, we talked about this before. Homozygous, you have to be specific about which one you're talking about because you can have homozygous dominant, where you have two big letters, and you can have homozygous recessive, where you have two small letters. Right, so you can't just say homozygous and assume that everyone knows what you're talking about. Now, on the other hand, you have heterozygous, which means that you have one capital letter and one lowercase letter. Another term for that is called a hybrid. So that's when Mendel took the tall plant and the dwarf plant, he crossed them together, and what he saw was all tall plants because the genotype was this, and the capital letter was the one that's in charge. So all the plants were tall. Now, in terms of genotype, we talked about this before, right? So your alleles are different combinations of the same gene, meaning you can have big T, big T, big T, little t, or little t, in this case. That's your genotype, so it's your genetic combination. And then you have your phenotype, which is the physical appearance. So whatever you see, right? You see tall plants, you see short plants, you see brown fur, you see black fur, brown eyes, blue eyes, things like that, right? So that's your phenotype. So again, hopefully you remember this. We talked about this before. So in terms of Punnett squares, again, I posted a couple of videos, and you are basically, in this case, looking at a diagram where you have 4 boxes. So it's a 2 by 2. So monohybrid, mono meaning one, hybrid meaning a combination, right? You're looking at one trait. So it could be fur color, right? So you can have black fur versus brown fur. You can have eye color, meaning brown eyes versus blue eyes. You can have earlobe types, attached versus detached, but you're only looking at one trait. So you're not looking at fur color versus eye color, right? That's not what we're doing. We're doing very, very, very simple Punnett squares. So in terms of Punnett squares themselves, right, we have a 4 by 4. So you have your boxes, right? And you have the different parents. So at the top, you have parent 1, right? So for setting up our chart, let's say here we're putting dad's gametes, and you remember that dad has sperm, those are the gametes, and then you have mom gametes, meaning the eggs. Here they're going down the side. It does not matter how you organize it. I could have put the 2 lowercase letters up top and the 2 uppercase letters down here. It does not matter. But what I did next was I basically took this big T, moved it into the box. I took this little t, moved it into this box. Right? I took this big T, moved it here, took this little t, moved it here. So I did this for all the boxes, right? And this gives me the genotype of the potential offspring, right? So this doesn't tell me that offspring 1 will look like this, offspring 2 will look like this, 3 and 4. This tells me that in one offspring, this is the probability that this will be their genotype. So from here, I can figure out the genotypic and phenotypic ratio, right? So if I'm looking at the ratio, I will tell you that it's 1 big T little t, and I will tell you that it's 1 tall, right? If there were different boxes, I'll show you what it would be like. For genotype, it's 100% big T little t. For phenotype, it's 100% little t. So for percent, it always adds up to 100. And then for the monohybrid, meaning one trait boxes, right? For those Punnett squares, everything should add up to 4. And if everything looks the same, you can technically say it's 4 big T little t, or you can just simplify and make it 1. All right, so in terms of monohybrid crosses, here we're looking at one trait. So we are looking at the dominant trait, which is green pods, versus the recessive trait, meaning yellow pods. We are looking at homozygous dominant for one parent, right? And we're looking at homozygous recessive for the second parent. So here we can see that 100% for the genotype will be big G little g, and then 100% of the pods will look green. So my genotype is 100% big G little g. My phenotype is 100% green. And if I'm looking at the ratios, again, you can do this in 2 ways. You can say 4 big G little g, and you can say 4 green, or you can just simplify that and say 1, meaning 1 out of 1 or 100%. So then if we are looking at further crossing, right, in this case, we're looking at plants of different colors. This isn't Mendel, this is just a different example, right? You can say if you have a homozygous dominant purple flower, right, big P, and you cross it with a homozygous recessive flower, meaning little p, again, 100% probability of the offspring will be big P or purple flowers, right? So this is similar to this, except we're using a different trait. Now, if you take these plants, and again, think incest, if you self-fertilize them, you will see that you will take 2 heterozygous plants, right? But in the F2 generation, you can see that now the original grandparent phenotype comes out. So again, I'm taking big P, right, heterozygous, and I'm crossing them together. Now here, if I'm looking at the genotypic ratio, I can see, it's right down here, 1 big P, that's right here, 2 big P, and 1 little p, right? If I'm looking at the phenotypic ratio, I can see that it's 3 purple to 1 white, and you convert those to percentages for if we ask you for a percent, right? 3 out of 4 is 75% purple, and 25% white, again, adds up to 100. And then here you have 25% big P, 50% big P, and 25% little p. Hopefully that makes sense so far. So if we're looking at monohybrid crosses, again, as I mentioned, you can have something like eye color, right? If you're looking at brown versus blue eyes. So over here, if you take your first cross where you have purebred brown eyes with purebred blue eyes, purebred means that it's homozygous, dominant or recessive, right? Based on what you just learned, you know that crossing this can only produce offsprings that are 100% big B, and all offsprings will have brown eyes. Now, I know that if I now take big B, you will always have a 3-to-1 phenotypic ratio, and you will always have a 1-to-2-to-1 genotypic ratio, right? If that doesn't make sense, make sure you work this out. So you have your heterozygous parents, right? You have, let's say, dad up here, mom down here. I fill in my Punnett square, so I take this big B, move it here, this big B, move it here, this one in here, this one in here, this one here, this one here, and these, right? So I distribute, and then I can count the boxes. So I can say that genotypically it's 25% big B. I can see that it's 50% big B, and 25% little b. Phenotypically, I see big B, big B, big B. So I know 3 out of 4 boxes, or 75%, have brown eyes, and 25% have blue eyes. Now, if I'm looking at this again, I'm counting the boxes for the ratio. So there's 1 big B, that's a unique genotype. There are 2 big B, that's a unique genotype. And there's 1 little b, so that's right down here. Now, if I'm looking at phenotypes, again, you have to be careful with those, because as long as there's at least 1 dominant allele, meaning 1 capital letter, it's going to look like the dominant phenotype. So in this case, 3 out of 4 boxes have a capital letter, so you have 3 brown eyes to 1 blue eyes for your phenotypic ratio. Again, hopefully that makes sense. If not, pose questions. So in terms of the practice problems, right, I saw some of your feedback, and I am looking at pages 1 through 11. Please work at your own pace. Please don't feel like you have to finish everything right now, but there are plenty of practice problems to get you to understand this. I'm also going to be posting a self-check mini quiz where you are answering questions about monohybrid crosses. So please look at this before, look through these problems, and then if you have any questions, message me, and only then do the self-check. So one of the questions you had was on monohybrid practice problems. It is question 2, and the question has multiple parts, right? It says, in rabbits, black fur is dominant to white fur. So automatically it's giving you a capital letter, it's giving you a lowercase letter, so you know what's dominant, what's recessive, right? And it's asking you for genotypic and phenotypic ratios, and what those offsprings will look like. So in the first example, we have a heterozygous rabbit with a homozygous recessive rabbit. Second example is we have 2 heterozygous rabbits, and then the last one is you have a heterozygous with a homozygous dominant. So I went through each one of these to hopefully help you with a simple example. So if we're looking at the first question, right, I know that a heterozygous individual, or rabbit in this case, is big B, so I'm writing that out. And I'm placing this parent right up top, right? My homozygous recessive is little b. I'm going to put the rabbit right on this side, right? So the first thing that I did was I wrote down the genotypes of the parents. The second thing that I did was I set up my Punnett square, and then from there, I'm going to set up my Punnett square and fill it in, right? So I take this b, move it down here, move it down here. I take this little b, move it here, move it here. Then I go to the second parent and I distribute the b's right across, right? So right away, I see that 2 out of 4 parents will have a dominant phenotype and 2 will have a recessive phenotype, right? So if I'm working this out, genotypically, again, we're looking at unique combinations of letters, right? there are 2 boxes out of 4 that have big B, little b. So I put 50%